This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1211, recorded on April 19th. No, recorded on April 18th, 2025. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi there, everybody. So what do we got here? We got a sunny and windy day. I don't have my weather app. Oh, here it is. Ooh, it says 91. So yeah, we're in climate change territory here. We've had more 90-degree days or greater than at any previous interval on record. But nevertheless, it's nice. We're good.
Here at 17C, it's sunny. It's really beautiful out. It's unbelievable. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It is 66 Fahrenheit, 19C, a little breezy, but a beautiful, beautiful day. I went for a walk after lunch. Gorgeous spring weather.
And from Madison, New Jersey, Brianne Barker.
Hi, everyone. Here it is also a beautiful day. My phone is saying 71 Fahrenheit, so 21 Celsius, and it's just lovely outside.
If you like these programs about science and the truth about science, please support us so we can keep doing them. Support means financial support. We don't do ads, if you noticed. Microbe.tv/contribute. Well, Kathy has not updated the ASV notes since last time, so I'll just tell you that ASV 2025, McGill University is hosting it. It'll be at the Montreal Palais des Congrès, July 14th through 17th. ASV.org/ASV2025, the ASV Cares and Kitty Core deadline. Yeah, Kitty Core. The deadline is May 9th for dependent care on site. Early bird registration ends May 12th.
That's Kitty Corp, not Kitty Core. You're not enlisting them in the Marines. They're the Kitty Corp.
Their name is short for Kitty Corporation.
Yes. They put the kids in suits and ties.
Got it.
Kitty Core was more like in Kindergarten Cop.
Yes.
Great movie.
Two news items. I'm sorry these are stories in the New York Times, which are paywalled, but you can find them elsewhere. Now, this week, the CDC, by the way, is still publishing Morbidity and Mortality Weekly Report, although apparently emerging infectious diseases is going to be shuttered. Can you believe it?
Right?
Wow.
Can you believe it?
I cannot.
Sure, I can believe anything.
Yeah.
Nowadays.
Anyway, this week in MMWR, CDC released the results of a long-going study showing that 1 in 31 8-year-olds were found to have autism in 2022, compared with 1 in 36 in 2020. Agency first started collecting data in 2000. And so they say, CDC says this is probably better awareness and screening.
Yep.
Right? But guess who says no, it's not? RFK Jr. said, nah, it's not. This is baloney. That's a canard, he said, to say it's better awareness. He said the autism epidemic is running rampant, which is also false, because Catherine Lord, a psychologist and autism researcher at UCLA, says it seems very unlikely it is an epidemic in the way that people define epidemics.
There was I don't know, I haven't read this article, so I don't know how much of this might be in this, but there was a report on my favorite news source, the PBS NewsHour, last night, covering this. And they said another reason for the change in numbers, let's call it that, is lumping together things that were not previously considered as being on the autism spectrum.
Yes.
into
Yeah.
autism. Like, for example, Tourette's syndrome, okay, which was its own thing before. Now it's in the autism bag.
Yeah,
Okay.
there are a number of things that were previously not diagnosed as autism that now are.
The category broadened and the awareness increased.
So here we are, folks, on TWiV, illuminating the truth. Go for
Well,
it.
you know, there's autism is an important one for us because it was originally said to be caused by vaccines, and studies showed that it wasn't.
Well, not
Yet-
originally, but at one point, because of a fraud
Yeah.
committed by a guy
Yes.
named Andrew Wakefield in an attempt to litigate, there was a flare up of belief in vaccines causing autism, which turns
then-
out to be completely wrong.
Then people said, okay, this could be a hypothesis, so we will do studies, observational studies, carefully controlled for observational issues, confounding issues like the Shingrix, the shingles vaccine dementia study, and they found no association with
Repeatedly.
autism.
In
Repeatedly, over
multiple
12
countries,
studies,
in
multiple
enormous
countries.
cohorts.
Yeah.
Yeah.
Yet we have people who cling to it and keep propagating it.
It, I've heard I spoke a lot to someone about this other story that you have, that these are kind of related, but I always found that comment about the relationship that you were just mentioning to be a little bit frustrating, in that even if it was true, I'm not sure that that's entirely a reason to not get vaccinated, because you're kind of saying that, like, you're calling a whole bunch of people in the world, like, you're saying their life is worse than measles. And that's kind of rude.
Yeah.
The second article I picked is also in the
Yeah.
Times. It's called Kennedy Claimed Autism Destroys Lives. Autistic People Disagree.
Yeah, and so I feel like I've said for a long time, like, I can't believe how rude it is that they implied that, and then yesterday he said it out loud.
Yeah.
And people got pissed.
Yeah.
He said, it doesn't destroy our lives. It's the ableism that does.
So,
So this guy,
So this spawns for me, or gives me a reason to do an out of sequence pre-pick. Have any of you guys seen the TV series Astrid?
yeah, I've seen it.
You've mentioned
You've seen
it, I
it.
think.
Yeah, it was a pick.
Maybe
of yours,
maybe I did pick it.
You picked it. Yeah, it's very
Okay,
good. It's very
awesome.
good.
Okay, so that really speaks to autism, really, and that
Right.
it's not, it's mischaracterized as being some sort of tragedy.
This lady who got autism at 17 said, Mr. Kennedy's comments are fear-mongering and his rhetoric flattens our existence into this outdated stereotype. He made these comments at a news conference on Wednesday. You know,
The
He
comments are awful.
they're horrible, and he just wants to build up momentum for him finding the cause of autism, right?
Which he says he's going to know by September.
Yeah.
Right.
I can't wait.
And you know what he's going to do.
No, he's already decided what the cause is, and he's just winding up for the pitch.
He is just going to collect bogus data
Yeah.
in a completely bogus way. And then I think he will cancel the vaccine, and we're going to be up the creek when that happens, folks. So you pay attention, folks, to what's going on. I mean, I don't want to get measles.
No.
What the hell? It's circulating because of this nonsense. All right. So we have actually 2 snippets today in a paper because they're all short. That's fine.
Oh, we
But
have
short- that
three.
short
We have
doesn't
3
mean-
snippets.
Short doesn't mean anything, actually.
Right.
Yesterday we did a TWiV, and Mark Martin said, "Oh, my paper is going to be 10 minutes, my snippet." And I said, "There's never 10 minutes because if it's interesting, we start to talk about it." It was about the isolation of a bacterium on the China space station. Did you know there was a China space station?
Yeah.
And so they isolated a bacterium, which originally came from Earth, obviously, but they got it off the walls of the space station. So it's growing there. And he said, "Oh, it'll take 5 minutes for me to." We asked him so many questions, it took a half hour.
Are
All right,
you
we don't-
sure it came from Earth?
Yeah, where else is it going to come from? Is it going to come in the door of the space station?
I think it came in the door. Yeah.
Yeah, it came through space.
Well,
It's
when they hook up, do they do spacewalks from the ISS?
Yeah.
from the ISS, yes.
So they open the door, but there's
Yeah.
a lock, right?
There's an airlock. Yeah.
Yeah, but the bacteria can sit in the airlock and then it can move into the. I don't know how it would survive. This is a run-of-the-mill bacteria that exists on Earth. It's a relative of Bacillus subtilis, you know. I mean, if you found something totally new, well, the thing is, the way they isolated it is they threw these swabs. This is funny. They took swabs and they swabbed the walls before launch and then at the ISS, okay? Not ISS, the China Space Station. Control was they took a swab and just waved it in the air.
Okay.
With or without the package. And they just put it into broth. You know, so if there's a bacterium floating around in space, it's going to grow in luria broth or whatever.
Right. Yeah, I mean, you could also imagine that maybe the organisms were on the suits of the astronauts or on something that came in.
Yeah.
Yeah, and then it establishes itself. It can grow in the walls. Yep. Anyway, it's never going to be short.
Right. So we have 3 snippets. Right.
Where do we get the third one? I thought there were 2.
There's a reply.
Oh, that's right. There's a reply. Yes.
Yeah. So our snippet group today, I can summarize if you like.
Really quickly because they're short.
Right, because otherwise, it's a triptych of comments in the latest issue of Nature about a paper we covered in TWiV 1131, mutant influenza H5N1 virus. That paper was about a clade of H5N1 flu virus called 2344B that's currently circulating in U.S. cattle and spilling over to dairy workers. You've heard about this. Now, avian-adapted flu viruses generally enter cells by binding surface sugars on cells that are terminated with an alpha-1,3 linked sialic acid structure. Human-adapted flu viruses bind to an alpha-2,6 linked sialic acid structure, and those are just 2 different chemical structures. It's generally thought that switching from binding from the alpha-1,3 to binding the alpha-2,6 is a key step for flu viruses to sustain transmission between humans, which is what we're interested in watching for. And in that original paper on TWiV 1131, Eisfeld et al. Was the paper, and they found that these clade 2344B viruses can bind alpha-2,6 linked, the human-type sialic acids, and I think they also did some ferret experiments to model human transmissibility, right?
Yep.
So now in Nature, this is a Matters Arising section, which is comments on published research. And it's 2 comments from 2 different groups who looked at avian H5N1 viruses with identical or, I guess, nearly identical hemagglutinin proteins to this clade 2344B. They found that these still, in fact, bind bird-type alpha-1,3 linked sialic acids. They don't bind very well to the alpha-2,6 human-like sialic acids, so they got kind of the opposite result. The 2 groups used several different assays to show this. Eisfeld et al. Followed up with a response to these 2 papers, and they compared and contrast the different methods that they used in their paper and the 2 comments. And it seems to me they conclude more or less with a shrug, saying basically, differences in the assays probably account for the different results. All 3 groups agree, I think, that we need to keep monitoring this situation, but it's unclear which way this goes right now.
Yeah, the original paper, which we did on the TWiV, so they had an isolate from a cow, an H5N1 isolate from a cow, and they found that it could bind both alpha-2, alpha-2,3 and alpha-2,6 sialic acids, right? Which, remember, the humans predominantly have alpha-2,6, although deep in the lung, they're alpha-2,3. In the eye, they're alpha-2,3. But you need to acquire alpha-2,6 binding if you want to be a pandemic virus. It's a job requirement. And so they said that it had dual binding, but the ferret transmission experiments, it poorly transmitted, which wasn't consistent with that. And we shrugged over that. We said it doesn't make a lot of sense, but who knows? And these 2 labs decided to re-examine it. And the links on these papers are weird. I couldn't download one of them. Anyway, one paper, or matters arising. I had to do a reader view, so all the authors are gone. Do you know the authors
Oh yeah,
of
yes, I can do authors on these.
receptor binding
Um,
specificity?
so
So-
am I on the first one?
Do you have that one?
Bovine H1. Okay, so the
There-
first one I got was bovine H5N1 binds poorly to human type sialic acid receptors.
Okay, but there's also receptor binding specificity of a
Yep.
bovine influenza A virus.
Shall
I've
I
got
do-
that one if you'd like the.
Shall I
those
do all
others.
the authors just in a row here?
No, you could just do the first and last.
Okay, bovine H. Well, no. I mean the paper. So bovine H5N1 binds poorly to human type sialic acid receptors. That comment is from 4 co-first authors. They are Jefferson Santos, Shengyang Wang, Ryan McBride, and Lauren Adams. Senior author is Scott Hensley, and
Who's
they
been on
are
TWiV.
from University of Pennsylvania in Philadelphia, Scripps in La Jolla, Francis Crick Institute in London, and University of London. The second paper is by 2 co-first authors, Pradeep Chopra and Sean Ray. Senior author is Geert-Jan Boons. They're from the University of Georgia, Athens, St. Jude Children's Research Hospital in Memphis, Ohio State University in Columbus, Washington University School of Medicine in St. Louis, Missouri, and Utrecht University in the Netherlands. And then the response is from the original group, and that I got to scroll through all these checkboxed things that came down with the paper. First author on that is Amy Eisfeld, and the senior author on that is Yoshi Kawaoka, and they are from University of Wisconsin-Madison, Heritage Vet Partners in Johnson, Kansas, University of Shizuoka in Shizuoka, Japan, Texas A&M Veterinary Medical Diagnostic Laboratory in Canyon, Texas, and the Texas A&M Veterinary Medical Diagnostic Laboratory in College Station, Texas, University of Tokyo, and the National Center for Global Health and Medicine Research Institute in Tokyo.
All right, so I'm going to do receptor binding specificity of a bovine influenza virus first.
Okay.
So they have an isolate, which is a clade 2344B from Ohio in 2024, and they use 4 different assays: Glycan array technology, biolayer interferometry, solid phase capture, and Glycan remodeled erythrocytes. Did you know you could take sialic acid off of erythrocytes and put others on? It's amazing. It's very cool. And they find no evidence for human receptor specificity of this bovine isolate. So all binding to alpha-2,3 sialosides. All right. Now, this virus they're using, the amino acid sequence of the HA, is identical to the one from the Kawaoka lab. Okay. So they use protein for, is it protein or viruses? Anyway, let me just go through it. It's the amino acid sequence of the HA is similar. They also have controls. They have a avian isolate from Vietnam, so that would be pre-cow. We have H1N1 2009 human pandemic virus, and the cow one binds, the Vietnam binds alpha-2,3, the pre-cow, and the 2009 binds alpha-2,6, and they use these to do the study. So first, the panel of glycans. So basically, you can synthesize all kinds of sugars, alpha-2,3 or alpha-2,6 linked sugars, and put them on glass slides. You can print them on glass slides, okay, and in very large numbers. So you can do hundreds or whatever. You can look at every permutation of sialic acid, either 2,3 or 2,6, connected to another series of sugars, because that could make a difference as well. And then what you do is you can put viruses on this microarray of sugars, and they do it in the presence of Tamiflu, which inhibits the neuraminidase. That's important because the neuraminidase would otherwise be cleaving the sialic acids off of the glass.
Yeah.
Right? And this was not done by Kawaoka. And they say that may be one reason for the different results. Right? And then, so they put the viruses on this array in the presence of Tamiflu, and then they determine binding using an antibody to the hemagglutinin stalk. And so the Ohio isolate binding only to alpha 2/3. That one had to be inactivated, right? Because otherwise you have to work on it at BSL-3. And the other viruses behaved as you would expect. The H1N1 bound to only alpha 2/6 and so forth. So they also showed that the inactivation didn't alter the binding preference. Okay, then biolayer interferometry. So this is a way of quantifying interactions, receptor ligand, if you wish, without any label. It's kind of like, what's the other one? The other technology that...
I mean, it's Biocore.
Biocore. Is that what this is?
Yeah.
This is Biocore?
I think this is Biocore.
I
Yes.
equate them.
So you put receptor on a slide and you pass ligand over it. And when the ligand binds it, you have a laser shining on it. It changes the angle of reflection so you can quantify the binding. It's very beautiful. It's just a very expensive machine, unfortunately. So anyway, they do that. So they have the sensors of the BLI. They're loaded with, these sugars. And then they dip the sensors into a buffer containing virus. And then, of course, they put it in the machine, and the machine measures the binding. So again, the Ohio cow isolate, the Vietnam 2004, and the California, they do all three of those in neuraminidase inhibitor. All the H5 viruses bound alpha 2/3, no binding for alpha 2/6, H1N1 bound only alpha 2/6. Okay? If you put just lactose on these sensors, nobody binds. Nobody likes lactose.
They're lactose intolerant.
Then a solid phase binding assay. So you have microtiter plates, a plastic plate with lots of wells in it. And you can put the viruses on the plastic. They will stick to the plastic. And then you can add the sugar polymers onto it in the presence of oseltamivir. And then you have a visualization of color as a way of measuring it. Again, the H5 only bound alpha 2/3. The H1N1 only bound alpha 2/6. And they did it. This is interesting. So if we didn't use Tamiflu, we got very low readouts, probably because the neuraminidase is taking the receptors away. Okay, now here's the cool part. So, hemagglutination involves adding influenza viruses to red blood cells. Red blood cells have sialic acids on their surfaces, and they will bind the red blood cells and agglutinate them, and you can measure that. And this is very funny. They said we developed a remodeling strategy to install specific glycan receptors on foul erythrocytes. Not foul, F-O-U-L, but F-O-W-L.
Yes.
Right? So they have turkey erythrocytes. They can treat them with the sialidase, which cuts off all the sialic acid. And then they can add back alpha 2/3 sialic acids using a sialyl transferase. I mean, it's not rocket science, but it's just enzymology, and it works. It's very cool. Because I guess red blood cells, surfaces are relatively simple. So they can add either alpha 2/3 or alpha 2/6, and then they do a hemagglutination assay. And guess what? Here they can use infectious virus in a BSL-3 or inactivated. But either way, the H5s hemagglutinate alpha 2/3 and not alpha 2/6, and the H1N1 alpha 2/6 and not alpha 2/3. And those are 4 assays, 4 formats. So there's bovine isolate, which has the same hemagglutinin as the Kawaoka, doesn't like alpha 2/6 at all. And they say maybe because of the neuraminidase inhibitor. But also, they used embryonated chicken eggs to grow their viruses, whereas the Kawaoka used MDCK cells, Madden-Darby canine kidney cells. Could make a difference.
Maybe different glycosylations.
Yeah.
On the virus.
I mean, so that's the thing, things can be different. You don't need much of a difference to make a different output.
I think what we've learned so far is that experimental conditions matter.
Yes.
By the way, that if you're wondering, the mammary tissue of cows has mostly alpha-2,3 sialic acids. That's what they say here. But the other paper, they say they have both. So
Yeah, right.
I don't know who's right because I don't study cows, and I never did. Did anybody here study cows?
No.
Yeah.
Nope.
All right. So anyway, so because here they say, okay, cow mammary gland, which is where this virus is reproducing, has only alpha-2,3. Therefore, opportunities for adaptation to alpha-2,6 are expected to be low. Yeah, if that's true. I don't know. I don't know if it's true or not. All right. So that's the one paper. And then the second one, just to be comprehensive, this is Scott Hensley's group who was on TWiV a while ago, remember?
Yep.
And so he did the same thing. He says here, in cattle, H5N1 replicates in the mammary gland, which expresses both alpha-2,3 and alpha-2,6 linked sialic acids.
So we have some disagreement on which sialic acids are in the.
So you can see
Cow
that
mammary
even
gland.
if an alpha-2,3 binding virus is reproducing in the mammary gland, it might say, oh, look, there's alpha-2,6. Let's try that out. And then you could select for viruses that utilize alpha-2,6. And that would be a pandemic potential because that's what you need to become pandemic. At minimum, you need to be able to bind alpha-2,6.
You need to be able to bind to human cells in other parts of the respiratory tract, not just the very deep parts in the eye.
Right.
Yeah, like your upper tract. All right, so here they made proteins. They made recombinant hemagglutinins, which are trimers. And they have an isolate from an American widgeon, which I presume is a bird, right,
Yep.
Alan?
It is a bird.
Not a widget, a widgeon.
A widgeon. It's a. Is it a duck? I think it's
No.
Yeah, it's a duck.
Is it spelled? It's spelled like widgeon, like
Wigeon.
like pigeon with a W.
Yeah.
Yeah, no.
Yeah, that looks like a duck.
They're pretty little ducks. Dabbling
Well,
ducks.
that's in 2021, so that would be pre-cow, right? And then they have a Texas cow isolate, and they have a South Carolina. So the South Carolina one is a WHO candidate vaccine virus, but it was made before the cattle outbreak. So I don't know which you'd be, if you'd use that or not. Who knows? Well, it depends. If this ever took off, you'd have to see the virus that's taken off and make the vaccine based on that. So they use 3 assays here. Binding to biotinylated glycans, but they're using just the protein, not the virus particles here. And again, the South Carolina isolate, the vaccine strain, only alpha-2,3. The Texas alpha-2,3, and the okay. So that's what's the human virus here? Did they say? Do they use one? I don't even.
No, I don't see one, and that's I got a problem with that. You need like the 2009 pandemic virus control.
Yeah.
To show that the alpha-2,6 binding is working.
That would be
All
good
right.
to see.
It would be good. Then they used a glycan microarray, like the one we just discussed. And again, these 2 viruses only bind alpha-2,3. And then they have whole virus, and they use biolayer interferometry again. And they find that the cow isolate from 2024 bound efficiently to alpha-2,3, but not 6. So it agrees with the other paper, controls notwithstanding, that these cow isolates are still binding to avian type receptors, contrary to the Kawaoka. And they say, differences in the assay, receptor analogs may be observed, but either way, we have to keep looking.
Yep.
So I do hope we don't stop surveillance because, so much other surveillance has stopped in this country. And everyone should know why. Because
Yep.
of funding and personnel cuts. I hope we don't stop surveillance for this. This would be so backwards. It would be ridiculous.
Just like I don't want measles, I don't want some kind of flu either.
No.
Well, measles surveillance apparently in Texas has been hampered because CDC doesn't have enough people anymore.
Yeah.
Right? So the thing is,
I don't want Widgeon flu either.
the cuts have not been thought of carefully. They have not been thought of carefully. And there's a phrase for this. David Gorski wrote this in Science-Based Medicine. He called it malignant incompetence. Right? You're incompetent, you make mistakes, and they're really bad effects of those mistakes.
Yeah.
I just see it on a daily basis. The dumbest decisions, not just with viruses, but we're not going to talk about outside of viruses, but all kinds of dumb decisions. The healthcare cuts and the research cuts and the university attacks, it's all malignant incompetence.
Mm-hmm.
Okay, that's the paper. Then we have Kawaoka, of course, responded as he should. And I couldn't download this one because...
Well, the whole Kawaoka group, so Eisfeld et al.
Yeah, of course. I couldn't download the PDF. But,
Oh, see, that one I didn't have a problem with. It was a different one I had a problem with.
I went to the press access door and got all these in one go. I was actually a little confused at first because when I downloaded the first of the 3 papers, they all 3 came down.
Yeah.
And then I downloaded the second one and I looked at the title. I said, no, I just downloaded that one. But then I realized it was the whole set was the same link.
Oh, it was the set. I didn't. Mine just came down as the wrong one. Anyway, so Kawaoka says, we stand by our results. Who knows what the differences are? But there are obviously some differences somewhere. And we still need to maintain surveillance. So my question for you would be, and I already know my answer. Would you go back and have these people all do it the same way to know what the results are?
I'd like to. I mean, it would be nice if Kawaoka's lab could do some of the assays that the other labs did, or vice versa. Just so that you're comparing.
Maybe they could share virus that was made in one place, or share proteins, or share whatever reagents.
Yeah,
so
so
they knew it was on the same reagent.
Kawaoka virus and MDCKs, he should give that to the other groups, and they could do it. Yeah. I don't know. I think we should just do surveillance and see what's going on in the cows on a regular basis. But obviously, we have to use assays that are going to give you a reproducible answer, right?
Mm-hmm.
Yes.
So if you do the next similar study, you don't want 2 out of 3.
The motivation then for the laboratory sharing reagents and doing that kind of stuff is good, be to figure out really what the appropriate assays are.
Yes.
Okay. So that when you go do this again, you're not confronted with the same confusion.
Yeah.
Right. This is how science works, folks.
Yeah, a nice big collaboration between all 3 of these groups would be good.
Yeah, and I'm sure they will, and they will.
And that happens. People do that.
If you read the articles, there's no nastiness. There's just these are the facts, and we don't know why it's different. There's no I'm going to take away your money because you got a different answer. It's ridiculous. Now, the second paper caught my eye, and it's in the Journal of Virology. Can you give us a summary, Alan?
Yes, I can. So this is the paper it's open access in Journal of Virology. It's stabilized dengue virus 2 envelope subunit vaccine redirects the neutralizing antibody response to all E domains. First author is Divina Thiono. Senior author is Aravinda De Silva. And the researchers are all from the University of North Carolina in Chapel Hill. So dengue virus, we've talked about this a number of times on TWiV before. It is a vector-borne virus that causes hundreds of millions of infections a year. It can be debilitating or deadly. And unfortunately, dengue vaccine development has been kind of a litany of failures, often pretty spectacular ones. So a major problem is that there are 4 serotypes of the virus. And in live attenuated vaccines, the 4 serotypes tend to replicate at different rates, which causes incomplete antibody responses against the different serotypes. Now, those partially effective responses can actually, in the case of dengue, enhance the infectivity of the wild-type virus. So subsequent infections could then be worse than if the person hadn't been vaccinated at all. So in the case of dengue virus, a little immunity is worse than none. Important to remember. These researchers are trying to get around that problem with protein subunit vaccines, so you don't have to worry about what replicates faster. These subunit vaccines use the E protein that's found on the viral surface, very popular target for these vaccines. People have tried this before, and they've found that subunit vaccines haven't worked very well. And then more recently, they've found that the E protein, when you manufacture it and just put it in solution, normally on the surface of the virus, it globs together in multimers. When it's just in solution and you're giving it as a subunit vaccine, it tends to be a monomer. So it's just individual molecules of E protein floating around. So in this paper, the team engineered dengue virus type 2 E proteins that form homodimers in solution to mimic the natural presentation of the molecule. And then they looked at mouse antibody responses to these engineered homodimers, and they found that the homodimers produce a much better neutralizing antibody response against the virus than monomers, and a kind of a broader antibody response that we'll be talking about. And then from here, the next steps would obviously be to design dimerizing E proteins for the other 3 dengue serotypes, because they did type 2 in this paper, and then test whether these subunit vaccines can actually protect against all serotypes of the wild virus.
So dengue, last year in the Americas, 10 million cases. So dengue can be asymptomatic. You can get dengue fever, which is an illness characterized by the usual symptoms, but also pains in your joints. We had a lady from Hopkins on TWiV a long time ago. She got dengue, primary dengue in, I think, working in Africa. And she said her joints hurt for months. And she said she would be fine. And then periodically she'd have these flare-ups of her joints hurting. So then, of course...
Yeah, I remember that. And last time I taught virology, I actually had a student who volunteered that they had dengue before. They weren't originally from the United States. And the student went kind of on and on about like, no, guys, you don't understand how bad it was. It was really unpleasant. It was really bad. And so I think that I was really struck by how much he was trying to emphasize to us how bad that joint pain was.
This is breakbone fever, right?
Yes, breakbone fever.
Because it feels like your bones are breaking. And this is consistent with this antibody enhancement issue. Often people who live in areas where dengue is endemic will get it. It'll be asymptomatic or minimally asymptomatic, and then they'll get another serotype, and it'll be horrible. So subsequent infection with other serotypes is a real danger, and they all spread.
You can get hemorrhagic fever and shock syndrome, and you could die. And the incidence goes way up after the second infection compared to the first.
As I'm listening to this and thinking about this, I'm recalling an episode we did a while ago on a paper that seemed to, at least in part, argue a little contradictory to this idea of the first encounter might be mild, second encounter worse, where there was an epidemiological study that said that in quite a few cases, or even a predominant number of cases, the first encounter was hemorrhagic fever. It was lousy. Now, I don't recall enough about that to know how that fits into the antibody enhancement thing. I don't think it refuted it completely.
The thing is that you do get some severe dengue after the first infection, right? And that study showed that it's actually a little bit higher than we think. At least I think it was done in India or some such place, but they found that it was higher than you would think. But it certainly goes way, way up in the second infection.
Okay.
Right? Maybe there are some people who make kind of a unique antibody response or an antibody response that's not quite as robust, and so they're getting that antibody enhancement a bit earlier.
Oh, that brings up, somebody asked me a question on stream Wednesday, and I said I would ask you. So if two people, unrelated, are given the exact same, say, peptide, okay, exact same amino acid sequence, will they make different antibodies against it?
Different antibodies?
Like with a different V variable region sequence? Or does it always have to be the same to match the epitope? Oh, this is great. I made you think a lot.
They could use different V's, D's, and J's to get to the same protein structure. It's sort of a convergent evolution kind of thing.
So if the structure is right for binding, the sequence doesn't have to be the same, right?
Right. And they're probably, in some cases, there's probably an easy way to get to that protein structure and a hard way. And so they might both be likely to do the easy way, but you will sometimes find somebody's B cells went the hard way.
Okay.
There's always somebody who's got to do things the hard way.
Exactly.
So these vaccines, the first dengue vaccines were infectious attenuated vaccines. First one was Dengvaxia, actually, where they took the yellow fever virus vaccine and they pulled out the structural proteins and just stuck in those from dengue, so type 1, 2, 3, and 4. And that was licensed into several countries, and it turned out that the type 2 response was very weak, and then that would be like a first infection to many kids, and then when they actually got dengue, it was worse. If they got dengue 1, it was worse because of the low anti-type 2. So you would make anti-type 2 antibodies that would cause enhancement because there's not enough to protect. And then another vaccine was developed, Qdenga. It's also been licensed, has similar problems. And as Alan said, one of the issues is that when you have 4 viruses together, they reproduce to different levels. You may get different levels of immunity. That was the problem with the polio vaccines, the infectious polio vaccines. You have to give 3 doses because at each dose, a different serotype replicates well. And then so with 3, you cover all 3 of them. So they say, okay, maybe a protein-based vaccine would be good. Now, the dengue virus is a plus-strand RNA virus. It's enveloped, and on the surface is, of course, the glycoprotein. But in contrast to all the other viruses we talk about, influenza virus, HIV, these envelope proteins are flat on the membrane. So, hemagglutinin is sticking up perpendicular to the membrane. But these are flat, and it consists of an E protein, which exists on the envelope as a dimer. And so they have made E protein-based vaccines. They're not very good. And they think one of the reasons is because when you make E protein, it's a monomer in these vaccines, and maybe that's not good enough for the generation of the right antibodies. And so separately, this group, I guess, has looked at the structure of the E protein and identified amino acid changes that would make the E protein, the soluble E protein, preferentially become a dimer. That's beautiful, right? And these have been shown not to affect antigenicity, these amino acid changes. So
to me, this is sort of conceptually equivalent to the making the changes in the spike protein.
Yes.
That stabilize it in the prefusion form. Because
exactly,
as I understand, correct me if I'm wrong, but as I understand it, first of all, this E protein does all the same stuff as these other proteins. 2 major functions: 1, binding; 2, fusion. Right.
Yep.
And the fusion is a property, but it's flat. Right. The fusion, in common with the others, there's a fusion peptide that in the prefusion complex is concealed because you don't want that thing sticking out and causing problems. You only want that when it's necessary.
Right.
So there is after the binding, a significant structural rearrangement in the dimer that exposes the fusion protein and makes the fusion happen. So much the same as the other proteins that we've talked about, this is a dynamic process. But in terms of making antibodies to the thing you want it in essentially the pre-fusion complex, or at least that's the theory here. And these stabilizing mutations that they've made do that. The pre-fusion complex being the dimer.
And that's why I picked this paper, because I think it's a neat idea, right? And it shows you this is basically what Jason McClown was talking about, vaccine engineering, doing things to modify the antigens that improve their immunogenicity. You don't just grow a virus and activate it, right? You do specific things. And so these changes that allow it to become a dimer, I think is brilliant, but it has to work, right? When you immunize animals, it has to give you the right antibodies. And what's interesting and important is that the two kinds of antibodies that are generated against E when you give animals virus or E, you get antibodies that just hit one E subunit. And then there's antibodies that bridge subunits, right? And those are both very important for neutralization.
Yeah, you got to do the testing because you want to make sure that your stabilization or whatever mutations you make don't also influence antibody reactivity.
Right.
So as Alan said, they're using dengue 2 E proteins. And what they're doing is immunizing mice. They prime and boost them with the wild type E protein, which makes monomers, and with a stabilized E homodimer, which is called SDFL. You can almost think of it as two prolines, right? The same idea. So you're modifying the spike to have good properties.
Yeah, it would be interesting to look back at their previous papers and see what sort of logic, if any, I'm sure there was some, went into identifying the residues that they had to change and how to change them in order to freeze this thing as a dimer. Probably has to do with, understanding what the structural rearrangements are between the monomer and the dimer.
You probably look at the interface between the monomers, the dimers, and then look at the amino acids and decide what to change. But I just want to mention, folks, that this is research, basic vaccinology research being done at a university. And if this works, a company will pick this up and push it forward.
Oh, yeah.
That's how, and the university is using government funds to do this. That's how science works in the U.S. It's a huge economic engine. Government, university, companies.
The company wouldn't do it on its own because they didn't know if it worked yet until they did these experiments.
Not only that, but if the particular pathogen and problem is sufficiently important as it was in COVID, the government may step in again to help with stuff like phase 3 trials, which are enormously expensive, beyond what many commercial enterprises are interested in doing under normal circumstances.
And just to continue on Rich's comment about COVID, the reason we had the infrastructure to develop a vaccine in 9 months and deploy it was because of decades of this university-funded research and government-funded research and this whole pattern that Vincent just described.
Yeah, so that vaccine required government, university, companies all collaborating. That's the beauty of science in the U.S., and that is exactly what's being broken at the moment. That system that evolved over 80 years is being broken.
And this was a system created by a guy named Vannevar Bush
for
World War II, and then continued after the war.
It's brilliant. Brilliant. We need another Bush, Vannevar Bush. Okay, anyway, back to dengue. So they immunize mice, prime and boost, with the two different proteins, with or without an adjuvant, which was alum. And just to tell you, in case you are interested, and there will be a quiz later. The SDFL antigen has 2 amino acid changes, not mutations. A259W, T262R. Those are amino acid changes in the E domain. 2 at the so there are 3 domains in this protein at the dimer interface. 2 amino acid changes in the hinge between 1 and 3, and a single amino acid change in the fusion loop. That all of them contribute to dimer stabilization. Interesting. So that 12 weeks, the mice immunized with the SDFL protein had more dengue virus binding antibodies, dengue 2, compared to animals immunized with the wild type. Right, so they can look at binding to the protein separately. There's another protein called M2P4, which has slightly different amino acid changes that also stabilize it and increase. Actually, those increase stability and secretion of the monomer. The SDFL immunized animals had higher levels of antibodies to both monomer and dimer antigens compared to wild type. So basically, this stabilized E protein induces higher levels of binding antibodies that favor binding to the homodimer versus the monomer compared to wild type E protein. So that's pretty cool. They also did a 2 versus 3 dose study in the absence of adjuvant because
it was so high with adjuvant.
Yeah, they also worried about the effect of the adjuvant on the protein structure.
Yeah, they worry that the adjuvant would essentially, partially at least, denature the protein. And we're talking about, we'll get into this, but the critical issue here is conformational epitopes, the way I see it. So the conformation of the protein is important.
So they did a 2 and a 3 dose study, and animals in the 3 dose group had higher levels of binding and antibodies, neutralizing antibodies. And in fact, I should have mentioned in the previous 2-week study, 2 dose study, they did binding and neutralization, which they do here as well. So 3 doses better than 2 in terms of binding and neutralization. The modified E protein SDFL stimulated antibodies in favor of the dimer over the monomer. So a 2-dose regimen is good, 3 doses is better. And in fact, 3rd dose actually improves the poor wild type E protein response, and you end up getting comparable levels of neutralizing antibodies for both groups. So that's interesting, but the answer is not to give the 3rd dose of the wild type. It's to try and modify it so that you get the right antibodies. The next, they look at the antibodies that are being produced, the 12-week high titer sera from the 3-dose study, and they're comparing the wild type and the modified E protein antigens. They found strong neutralization of dengue 2, but not the other serotypes. That's good, because they didn't use the other serotypes.
That's right.
But also shows you that there's no cross-reactivity, which is very interesting.
Yeah, and it's an important control too, because you can do these kinds of things and end up with something that just kind of gooses the immune system really well. And then you boosted immunity against everything. And so doing this control with additional serotypes and saying, nope, we're specific, that's important to see.
Well, and it's interesting because you could sort of imagine either answer being kind of interesting.
Yes.
If they had cross-reactivity against all the serotypes, then you'd say, yay, we now have this amazing vaccine that's going to protect against all serotypes. You know, we did it. But we also know that part of the problem with antibody enhancement is sort of this weak cross-reactivity. So we either want like really good cross-reactivity or none.
Yes.
And here they see none.
So they want to know where these antibodies are binding on the E protein, right?
And I would like to comment, if you did see that the other serotypes of dengue produced really good antibodies, I would also want to do then an experiment with some totally unrelated virus.
True.
And see if we've just, boosted all the antibodies, which may not be a great vaccine approach. But yeah.
So to do this, to map the binding sites of the antibodies, they look at a panel of recombinant viruses between dengue 2 and dengue 4. Right? They're going to substitute parts of the E protein of dengue 4 with pieces from dengue 2, because remember, the antibodies are not binding to dengue 4, so that'll tell you when you move a piece of 2 over and it reacts with antibodies, that piece has the antibody binding part. Okay, and so they find that both wild type and the SDFL antigens make antibodies that will bind and neutralize a virus that has the E domain 3 from 2 into 4. So these you can see that both are inducing antibodies that target domain 3 and also antibodies that span 2 and 3. The modified E protein also make antibodies that neutralize ED1 and 2 chimeric viruses, whereas the wild type protein made lower levels of neutralizing antibodies that mainly tracked with binding to domain 1 and no detectable neutralizing antibodies to domain 2. So basically, wild type protein is mainly inducing domain 3 targeting antibodies, while the SDFL induces antibodies that are directed against all 3 domains. And I presume that's what happens in people as well, and that's what you want, right?
And I'm been a little distracted here because I'm trying to look at the structure of this protein and see if I can understand where in these graphics each monomer. I assumed initially that actually in this one that labels the domains ED1, ED2, and ED3, I think the labels are all on one monomer. I originally assumed that each monomer was like one straight across the top, one straight across the bottom, but I don't think that's true. I think they turn the corner here. At any rate, that's the important thing is that they talk about one of these domains comprising it basically being a conformational domain, as I understand it. That is, it would exist only in the dimer and not in the monomer. Do I understand that correctly?
Yeah.
Okay, so that to me is the kind of the bottom line on this, is that in order to get that conformational, an antibody made to that conformational epitope, you need to have it in the dimeric form. The monomer, you don't have that epitope present in the monomer.
Right. Okay, and the last experiment is, as I mentioned before, so some antibodies that neutralize the virus map within a single domain, and others bridge E domains, right? On the same E or on different E's. So within the monomer could bridge different domains or across E domains. And they have actually antibodies, monoclonal antibodies that mimic those, and they know where they bind. And so they want to know the contribution of simple antibodies to neutralization after vaccination with the wild type and the SDFL antigen. And so what they do is they immunize the mice, they take sera, and they incubate them with dengue 2 domain 3, E protein domain 3 coated beads, and remove all the antibodies that bind just domain 3. Remember, that's what the wild type protein only induces, just mainly against domain 3. And removal of these antibodies, domain 3 antibodies, led to a reduction in binding to dimers, which also include simple epitopes. And then they looked at neutralization. So when you remove the E domain 3 binding antibodies, the neutralization drops below the level of detection of the assay. So you're basically not neutralizing anymore. And so the removal of these E3 binding antibodies also lowered, that's the result I just told you with wild type E protein. If you do the same experiment with animals immunized with SDFL, there's a fraction of antibodies resistant to depletion by the mesh that I told you. So obviously, those are targeting different domains on the E protein. So they say this is further proof that this SDFL antigen stimulates neutralizing antibodies against all 3 domains on the E protein.
So they basically see that a higher fraction of the antibodies stick around.
Yeah.
So it seems to me that now what I would do is to take out individual B cells and sequence some antibodies and make them and see where they're binding. right? Because
Yes.
these assays are kind of broad assays, but if you get individual antibodies, oh, this one is binding to just domain 3, and this one is bridging, and then you can compare the two antigens. I think that might be interesting or informative. Not interesting, but informative. Okay, sorry, you were going to talk.
I was going to say exactly the same thing. And I was going to, sort of be looking at Brianne at the same time and asking
She
what
was
she
nodding.
would,
She was
what-
nodding,
what
so
would
I
you
thought
do?
it was okay.
And
I
I'm
think that
wondering
makes sense.
wondering how difficult this might be, because you're basically talking about fabricating a bunch of essentially monoclonals
Yeah.
after all these immunizations, and looking even directly structurally at how they bind. Okay, and looking to see what sort of epitope specificities arise from a structural point of view
Right.
in each of these cases. Not an easy job.
No, and really, I think the goal of all of this, the goal of doing those experiments would be to give you more insight onto what is the right immunogen, because you'd find out an epitope and then you'd say, okay, how do I design something complementary to that to be the immunogen? And this paper is already showing us a pretty good strategy for making an immunogen.
Yeah.
So as I read this, I think about sort of the evolution of vaccinology, and I'm probably overgeneralizing here, but this is my impression, having lived through a fair amount of this, is that live attenuated vaccines have been a big deal for a long time as being sort of the most effective way to do things. But they do have some downsides, as we've discussed repeatedly. And it seems to me that with the advanced technology that we're getting, advanced understanding of the immunology, and the advanced technology we're getting with achieving with protein engineering and stuff, that there's an emerging science of protein vaccines that are every bit as good and ultimately maybe even better than the live attenuated vaccines, because they don't have any of the downsides. Okay. Am I talking sense?
No, you're totally talking sense. And I think that, one could sort of debate how much of it is a protein antigen or protein immunogen versus a nucleic acid immunogen, encoding those proteins,
Sure.
and why you'd use one versus the other. But in any case, I think we're getting to the point where we can use those types of technologies and get awesome immune responses that don't have the downsides of the live attenuateds and don't have the risk of the live attenuateds.
Where some of this breaks down, I suppose, but it'll come full circle, is it could be that a lot of the best immunogens that come with live attenuated viruses involve more than one antigen. Okay, and so you really need the whole virus, or you need several gene products.
It might be more than one antigen, or an antigen plus an innate immune stimulator,
Yes,
or thinking
right.
about what the dose is,
Right.
and whether the live attenuated is getting you to that dose. But those are all by replicating. But those are all things that some of our new technological approaches let us address.
And in terms of some of the kinds of protein engineering, I think there's been an evolution there as well. Sort of the first case that comes to my mind is the papillomavirus vaccine, and several others that are virus-like particles, okay, where the theme is the same. The individual monomer antigen doesn't work very well, but if you make a virus-like particle that's more likely to have all the conformational epitopes and that kind of stuff, that works. And there are several examples of that kind of thing. And now it's and that includes nanoparticles, right? Where instead of making a virus-like particle, you make a cluster of antigen by hooking them all together in a nanoparticle somewhere, and that comes close to the same kind of thing. I have wondered for a while, and I don't think we've ever done a TWiV on this or looked at any papers about the Shingrix vaccine. Because making vaccines to herpes viruses, as I understand it, has been problematic for decades. And so the first chickenpox vaccine was a live attenuated vaccine. And that has been replaced by basically replaced by a protein vaccine called Shingrix. And I don't understand what the magic of Shingrix is. How is it that we got this chickenpox or varicella zoster vaccine that works as a protein vaccine? We're, I guess, still having trouble with some of the other herpes virus vaccines and couldn't do this before with varicella zoster. What's special about the Shingrix vaccine? I don't know.
It's a good question. By the way, the kids still get the original.
Okay,
chickenpox
the original
vaccine.
guy, not the Shingrix. Okay.
But I believe it's Shingrix is being trialed in kids to prevent chickenpox.
Yeah,
because
I mean,
it's so good.
that would also make more sense, because the problem with giving kids a live attenuated chickenpox vaccine is you are making them predisposed to shingles later on by giving them that latent infection potentially. So if you could switch to the protein vaccine, that would be better. I think there's something weird about the adjuvant with Shingrix, but I don't know the details either.
Maybe we'll get mail.
Yes.
We could even do some research, but
We could.
So here we have a modified E protein of dengue 2, which gives you higher antibody levels than the unmodified E, because it's a dimer, not a monomer, and also antibodies against all 3 of the domains of E that are neutralizing. So this, I presume, should go forward.
Yeah.
I don't know what would be next.
Well, I think next would be the other serotypes.
Exactly.
Other
That was my
serotypes,
thought too.
yeah.
And then from there, combining them and putting them into first mice and then non-human primates, and seeing if you can work out. Probably you're going to have to do some tinkering on the dosages of the different ones.
Yeah.
And it may be like a polio vaccine case where you need to do repeated immunizations so that you get, antibodies against the different serotypes.
Did we mention that, because I was distracted for a while, did we mention that the immune response you get to this particular thing is highly specific for one serotype?
Yes.
Yeah.
Okay.
So they also mentioned doing this with Zika, which is another flavivirus, which would be interesting. So this is cool. I like this very much.
Yeah.
Very interesting. I was worried about the attenuated dengue vaccines, and this could solve the problem. Yeah. All right. Do some email today. Rich, take the first one, please.
Harold writes, "Hi, TWiV team. Regarding your recent banter on German humor, this was somebody, some email talked about Southern German humor, and we were wondering what that meant. Okay. Regarding your recent banter on German humor, I can't speak to what differentiates Northern versus Southern German humor, but I can share a joke. Here's the joke. How many Germans does it take to change a light bulb? 1. We are very efficient and do not care for humor. That gets a laugh around here, but maybe that's just Midwestern perception of the ancestral homeland. Cheers, Harold. Great. Thank you, Harold.
Nice.
Brianne, you want to take next 2 or so?
Sure. So Anthony writes, your Aunt Sadie, who sells on eBay pharma, corner candy store pharma, to replace big pharma. And so Anthony gives a link to a Politico article about some changes that may happen with big pharma, and says, for what it's worth.
So this is a visit. So RFK visited the FDA, and he said there, this is in this article, that the deep state is real and the FDA employees are sock puppets of industry. I happen to know at least one FDA employee.
I was going to ask.
Say that person is not
No.
a sock puppet of industry.
The idea of anyone describing that person as a sock puppet just does not fit.
Someone very much of her own opinions.
Helen, how can you get up in front of an organization and tell lies? I just don't understand this.
He doesn't perceive what he's saying as being lies. He's in this alternate reality.
So he fired 10,000 employees last week. Oh, he said, we'll do more with less. I really have confidence in him. Oh, boy. Anyway, that's that article. Sorry, go ahead, Bria.
No, that's fine. And then Brenda writes, I'm sure you've seen this, and gives a link to an MSN article about RFK Jr.'s plan to set up a study to determine the cause of autism by September. And Brenda says, shouldn't he be commissioning a survey of all the scientists currently researching the cause of autism before adding new ones? I wonder how many of the current ones have had their funding withdrawn. Maybe they should lawyer up and make a fuss. Yeah, I agree, Brenda. I have questions about some of the folks who are involved in this new study, and I would much rather that those who were previously looking at autism stuck with it, with the funding that is being used on this.
So people have been working on understanding the causes of autism for many, many, many years. How can you say you're going to figure it out by September? Oh, it may be that you don't know how science works, right?
Yep.
Science doesn't work by you saying, I think there's an environmental component. I'm going to find it. That's not what you do. No. That's a bias. You're supposed to say, what's the cause of autism? And examine many things. And many things have been examined. You know, there's a genetic component. And he in this article says, genes can't cause epidemics. That can't be right. Really? Really?
Well, question number one is there an epidemic?
Right.
Well,
The premise there is flawed.
Yeah.
Yeah, that previous article in the news, the lady said, I wouldn't characterize this as an epidemic, right? It's endemic, I guess, if you want to use that term. Maybe they should lawyer up and make a fuss. Boy, the lawyers are going to be busy the next couple of years.
Yeah, the lawyers are making out like bandits, but that's fine.
It's okay.
Some of them are doing us a favor.
Okay, Alan, you're next.
Sure. Joanne writes, Dear TWiV, I was born in the 1950s and had chickenpox as a child. A few years prior to COVID, one of my close co-workers came down with chickenpox. A few weeks after he became ill, I developed some blisters on my arm that strongly resembled those that caused by chickenpox. Fearing that I might be contagious, I went to the doctor to see what should be done. He confirmed that the blisters indeed looked like chickenpox, especially given my recent exposure. He advised to work from home until the blisters cleared up. I never felt ill, no fever, etc. Only symptom was those few itchy blisters. My poor co-worker was not so lucky and was extremely ill for weeks. So now I'm wondering, why did I get a mild case of chickenpox rather than coming down with shingles? Is there a difference based on re-exposure to the virus versus reactivation of the virus? Is this any different immunologically? This incident did prompt me to get the Shingrix vaccine. I should point out that I never had any lab tests to confirm infection. Diagnosis was based on history of recent exposure and visual observation. Keep up the good work at TWiV. These are dark times indeed. Okay, Joanne, I can take a little bit of this. The diagnosis not being confirmed, I mean, you don't know that you had chickenpox, basically. So this could have been something else. This could have been maybe you brushed against some poison ivy. I know it looks a little different, but maybe this was some other problem that came up, caused a rash. But if it was chickenpox, that can happen. You could have incomplete immunity to it. And I am guessing that I think I recall something about this. The reactivation and the infection process are proceeding differently.
Yeah.
In reactivation, you've got the dormant virus coming out of a nerve ganglion, coming down the neurons and going to the site where it's going to erupt into shingles. In infection, you have the virus going in from the outside and infecting and causing the rash. So they are different disease processes, and I suspect that's the reason. What triggers shingles is not usually an exposure to chickenpox.
And usually isn't the first infection respiratory?
Yes.
That's right. Yep.
And I'm thinking that the immunology involved might be different as well.
I'm sure that it is, just based on the differences in cell types and sort of locations, but I don't know a ton of details on that.
The next one's from David. Dear team, keep up the great work. With respect to vitamin A, anti-vax, RFK Jr., vaccine deniers et al., aren't they simply demonstrating both Darwin's natural selection of the fittest and Lamarck's inheritance of characteristics acquired from one's parents. Think about it before you discuss. We have vastly overestimated our culture, alas. I don't see the connection between all these dumb things and selection of the fittest, right? Because we have medicine now that keep people alive, even if you do dumb things. Does anyone have a better viewpoint?
I'm not sure where he's going with this.
I'm not either.
Yeah, I mean, I think part of it is that, yes, you are acquiring, some of this is coming from your parents because kids are the ones who are impacted.
Yes,
but they're
yes.
getting impacted because of the beliefs of their parents.
Yeah.
But I'm not sure that right now there are enough situations where people are not getting medicine to really be seeing a difference related to Darwin or Lamarck.
So we go backwards to pre-19th century when the concept of science and scientists gained its first traction, according to this book by Paul Craddock, Spare Parts, The Story of Medicine Through the History of Transplant Surgery, May 2022. I haven't read this book. The History of Transplant Surgery goes way back. I see. So I don't know the connection there either, Paul or David. Does anyone else?
No.
Okay. All right, we have one more. Who had gone first? Rich. Okay, it's your turn, Rich.
Okay, hang on, I was distracted.
So basically, Rich, she is quoting an article here. You could choose parts to read if you'd like.
No, go ahead. You've obviously looked at it. Go ahead.
Okay. This is Anne. Anne writes, finally, from Barron's. Two top Wall Street analysts are calling on the White House to remove RFK Jr. as the U.S. Secretary of HHS, saying he is undermining public health. New research note from Cantor Fitzgerald. Biotechnology analysts Josh Schimmer and Eric Schmidt was spurred by the surprise resignation of Dr. Peter Marks, a top FDA official responsible for regulating vaccines. The analyst said Monday that the forced departure of Marks, who led the FDA's CBER, had been a step too far for us. RFK is undermining the trusted leadership of healthcare in this country. HHS cannot be led by an anti-vax conspiracy theorist with inadequate training. And they go on more. I just want to point out in his resignation letter, Mark said he'd been willing to work to address Kennedy's concerns about the safety of vaccines, but that Kennedy wishes subservient confirmation of his misinformation and lies. I thought that was really good. Knowing Mark's, not knowing him personally, but I've seen him talk a lot. He's a very conservative guy. For him to say that, he's got to be pissed off. Let's see, what else is worth... Anyway, so if you care about the industry, if you care about healthcare in America, you should be very, very disturbed by what happened. Which is Mark's being fired, I guess.
Yeah.
Well, I think it was that he resigned because he was so over it. He wasn't fired.
Well, they said to him, you either resign or we're going to fire you.
Oh, well.
So they take that as a firing.
To me, this all speaks to the question that many of us have been asking for 10 years or so, which is how bad does it have to get before people wake up? Yeah. Maybe we're going to find out. And unfortunately, it looks like it has to get pretty bad. But, it comes down to the sort of individual population experience, I think, that, enough individuals who are just trying to live their lives have to be negatively impacted by this kind of stuff to figure out that it's not good.
Yeah.
So, unfortunately, in order to influence enough people to make a difference, it's got to get pretty bad. We'll see.
Yep. Okay, let's do some picks. Brianne, what do you have for us?
I have an article that I saw on CNN earlier this week. That's called "Scientists Detect Signature of Life on a Distant Planet," study suggests. And so this is a report on a paper that just came out in the Astrophysical Journal Letters, talking about a planet called K2-18b, where the James Webb Space Telescope was used to detect certain chemicals within the atmosphere that the authors say are on Earth only made by phytoplankton, marine phytoplankton. And so they are hypothesizing that there are there are organisms that are making these compounds on this planet, which I thought was super cool and super exciting. But I also would say that this, and it talks a lot about how the Webb telescope has been helpful and things like that. I also kind of liked that this article had a lot of information from people who were on the other side as well. It had a bunch of information from skeptics kind of explaining what they thought the same data meant, what observations or things that they kind of were imagining. And so it kind of described both sides of, this is the discovery, this is what it does mean, this is where faults are, in a way that made it, that I didn't always see in a lot of press articles about science, because it really kind of described the process and the things people were looking for and the things they might do next. And so, I will fully admit that when I saw the article title, I clicked on it and was super excited to read it, because this is a type of thing I like geeking out about to read. But one of the reasons why I ended up liking the article so much was it was really interesting and cool as a topic, but I liked how it covered sort of the back and forth between the two different sides.
So what are the data? Is this some sort of spectral hit or something like that?
Yeah, so basically they are saying that they interpret some spectral data as being dimethyl disulfide or dimethyl sulfide.
Okay.
And they say that basically that the idea of having those compounds in a world like this one, which is a world that they say is covered in liquid water, would only work if there were organisms creating those compounds. It turns out that other people don't actually believe that there is necessarily liquid water. So there's debate about what the composition of the planet looks like. And then others don't necessarily think that the spectral evidence is quite saying what they think.
Is there discussion of the environment being a reducing environment or something like that, or an oxidized? Because disulfide bonds, right? Those are
right. They don't talk about reduction and oxidation in this article.
Okay.
They talk more about the temperature, because some think that there's sort of this sort of water, but while others think there's a magma ocean. And so those differences in temperature,
Kind of an important difference.
Yeah, those differences
magma doesn't strike me as a great place to live.
Yeah, and so that was the bigger thing that they were talking about, was a difference in temperature, which would involve difference in how hospitable the planet might be.
And it's 124 light years away, so you wouldn't want to go there and then find out that it's magma.
Yeah.
This is all based on the fact that on Earth, DMS is made by microorganisms.
Yes.
Right, exactly.
So,
and so they're assuming there's no other way for it to be made,
yeah,
which we
be
certainly
right.
also don't know. But I just thought it was cool. And I said,
yeah.
I liked that it talked about both sides.
And it's really, really awesome that it's even possible to have data that would spur this discussion. There's 124 light years away. This thing is way out there, and it's a planet around a star, and you've got telescope data that's giving you enough information that you can talk about atmospheric chemistry. Wow.
Yeah, it's super cool.
Rich, what do you have for us?
Okay, so a little preface here. I've been thinking a lot about motivation recently, in particular in the context of this podcast, because we say we don't want to get political, right? But it's hard to avoid in the current atmosphere. And so in my mind, I've been kind of saying that I was going to draw a line for myself at reporting or discussing what facts were and stopping short of discussing motivations, because we don't know what people's motivations are, okay? And I keep a file of stuff that might serve as TWiV picks. And so this is rummaging around in my mind, and I'm going through my TWiV picks, and I come across this thing called Hanlon's razor, which actually I think speaks to motivation, or the question of motivation. So I'm going to say this is the part of the podcast where we can talk about at least flavors of motivation. So Hanlon's razor, I don't know where I got this, how I came up with this. And this is a Wikipedia entry. And just to read from that entry, Hanlon's razor is an adage or rule of thumb that states, quote, never attribute to malice that which is adequately explained by stupidity. Okay? I love it. And then it goes off on all kinds of stuff. And this really gets me thinking. Now, the interesting thing is that whether something happens as a result of malice or stupidity doesn't really make a whole lot of difference. The phenomenon is the same, okay? It's only, the motivation that got you there that's different. There's some exceptions.
Yeah, I like some of these other versions of it.
Yeah. Gray's Law. I like this. Any sufficiently advanced incompetence is indistinguishable from malice.
Yes.
Okay. So I don't know. I don't know what the motivations are and I should say, and I've talked about some of this stuff before. I have spent time in my life studying Buddhist philosophy, and a major sort of issue in Buddhist philosophy, I don't know, or concept, is what's called the three poisons, which I've mentioned before. And these are the things that ultimately give rise to craving and which gives rise to suffering, okay? So these are the things that make us miserable, okay? They're innate characteristics. And they are greed, hatred, and delusion. And it's really very interesting, because if you think about this, spend some time thinking about this, it kind of covers everything in a non-overlapping fashion. The Buddhists really have thought about this stuff very carefully. And as I look at that and compare it to Hanlon's razor, I would equate malice with hatred. And I would equate stupidity with delusion, and say that the motivation here that's missing is greed.
Malice could be greed sometimes.
Malice could be greed.
Malice
sometimes.
can be greed.
Okay. At any rate, if we want to think about, in our spare time, motivations for all this kind of stuff, I think Hanlon's razor is worth keeping in mind, as are the three poisons. I'll summarize that way.
I mean, let me speculate just a bit. So we have this conservative Christian document, this 800-page thing, 2025 plan, right? Remember that thing?
Yeah.
Which is now basically being played out.
Yep.
All the things in that we see happening. That arose because a bunch of conservative Christians in this country feel they're being ignored, and this is not the way the country's. They're being stupid. Okay. Instead of negotiation and discussion, they want to trash. They think that's the only way to train things, and that's stupid. So that's the motivation, in my view, for all this crap that's happening. I could be wrong, but that's how I see it.
Well, I think one of the things that happens is that lots of different individual motivations wind up getting swept up into that giant brew. Okay, so individuals may have their own specific. Actually, there was another TWiV pick in my pot that I almost picked out. I've got it here. An article from a year ago titled, this is from the Washington Post, saying tax records reveal the lucrative world of COVID misinformation. Okay.
Yeah.
So this is where the greed motivation comes in, and it documents how during the pandemic, numerous COVID misinformation organizations, including, or most notably, the Children's Health Defense, which is a vaccine group founded by RFK, their donations increased many-fold, as did the salaries of their CEOs, as did donations from conservative groups during the pandemic. So,
Yeah, it's a very interesting thing, Rich. I appreciate that. Okay. Well, there's another one. Remember the Great Barrington Declaration?
yeah.
What's the motivation for that? Stupidity. You want to let it rip. That's basically let it rip.
Also, greed.
Yeah, I was gonna say that was
Greed.
greed.
Greed
Yes.
was a major motivator of that.
Yes.
Let's
in
reopen
fact,
all the businesses so our
Yes,
stock portfolio will
exactly.
go back up.
Yes.
And
You're
we're just going to
absolutely right.
rationalize that.
Don't make me lose money by having my business be
Yes.
closed.
And that's why one of the authors was Jay Bhattacharya, who's a
Yep.
health economist. There you go.
This is great. So from now on, when we get around to the motivation question, we can subcategorize motivations
Ascribed it, yes.
in the terms of the three poisons. Is this
Yes.
greed, hatred, or delusion? Or all of above?
Or all of the above.
Alan, what do you have for us?
I have something that I think is just fun and cool. It's kind of like Brienne's pick, but underwater. So this is, I picked an article in Science. I think this video has been online a few different places. This is an undersea survey. They're using remote operated vehicles in the deep, deep ocean and looking around at stuff. And they captured actual footage of a colossal squid in its natural habitat for the first time. Now, before you think of, the squid eating the Nautilus in for 20,000 Leagues Under the Sea, this is a baby. So it's actually a little tiny life form, but the thing grows, this species grows it can weigh half a ton and grow to 7 meters, like 20 feet. It's the biggest squid in the ocean. And until now, all we've known about them has been from individuals that got caught up in trawl nets and brought to the surface, and they're all damaged, or the remains of these that are found in sperm whale stomachs, because sperm whales dive down there and eat these giant squid. But this one is actually swimming along, and it's beautiful. It's translucent and just moving along with its tentacles behind it, and it's video of the thing. It's just really cool.
So 600 meters down, right?
Yeah.
1,800 feet.
Yep.
That's pretty deep.
Crushing.
That's deep.
Pretty crushing.
Yeah. The heaviest known invertebrate.
Yeah.
Cool.
But how does it taste fried up in olive oil and butter?
I want to see its viruses.
Yes.
That's it.
There must be, right?
Mm-hmm.
All right. I have a pick in a Nature article. Unfortunately, it's an essay, and it's behind a paywall. It's by Steve Blank. How the United States became a science superpower and how quickly it could crumble. Oh, this is such a good article, folks. Read it. U.S. global dominance in science was no accident, but a product of far-seeing partnership between public and private sectors to boost innovation and economic growth.
I don't think this is paywalled.
I'm popping right up for me.
I clicked to
that's
accept
good.
cookies, and I can read the thing.
Oh, good. Collectively, universities spin off more than 1,100 science-based startups each year, leading to countless products that have saved and improved millions of lives, including heart and cancer drugs, mRNA-based vaccines. These breakthroughs were made possible mostly by a collaboration between U.S. government and universities, which, as Alan said, was started actually during the war and to make weapons, and then afterwards used to make good stuff. And they go through numbers. I want to just tell you what some of the numbers are. The U.S. government is the largest source of academic R&D funding globally. Its budget is $200 billion in the year 2025. More than 24 agencies give out that money, including NIH and NSF, right? Those are the two biggest ones. And he says the agencies do this for a reason. They want professors to do their research. In exchange, The agencies get research, which moves science forward. By partnering with universities, the agencies get more value for money. This is because universities can leverage investments from the government with other funds. In 2023, universities received $27 billion from charitable donations, $6 billion in industrial collaborations, $7 billion from nonprofits, $5.4 from state and local government. So that boosts the NIH $59.7 billion up to $100 billion. So, this is an ecosystem. It's the engine for creating startups and jobs. In 2023, universities licensed 3,000 patents, 3,200 copyrights, and 1,600 other licenses to technology startups. This is an amazing engine, an economic engine. It's being destroyed right now. This government university industry triumvirate is being destroyed. I think it's greed.
I think it's greed too. If you didn't know what we were talking about earlier with Vannevar Bush, this also explains that.
There's a heavy dose of delusion in there.
I think I was going to say, Alan.
Whole lot of that.
So delusion in the fact that they think that these things are all corrupt.
Yeah.
There's this. Who is it? Russell Vogt was on the news the other night talking about, and this was about the recent Harvard dust-up. Yeah. Okay, talking about the universities being basically the source of radical liberalism.
Yes,
okay?
Exactly.
And so there is an effort to sort of stifle that. Okay.
Yeah.
So
sure.
I would put that. I think
I'd put that in the hatred category.
That's a sort of a crossover between malice and delusion. Okay, because first of all, it's not true. Okay. Well, we could get into that discussion, but it's not like universities are designed to teach radical liberalism to anybody who goes there. So I think there's some delusion there. There's also some malice. I don't like this, so I want to get rid of it.
Well, there's no question that the universities are where free thought can occur. So you don't have to be a liberal, but if you want to be, you can go and you can be an activist or whatever you want. And the conservatives hate that. They hate it. They have no control over it. And even more important, the universities and colleges know when a despot is speaking BS. And they speak out against it, and they don't like that. So you silence the universities first. It's quite clear.
Yeah.
But that's part of this great triumvirate that's an economic engine. So you're really shooting yourself in the foot.
I wasn't going to talk about motivation.
Yeah.
Anyway, it's a really good article.
Yeah,
Take
this
a look
looks
at
great.
We have a listener pick from John. Thought you and your listeners would be interested in the most recent podcast from Malcolm Gladwell. His Revisionist History podcast usually ends with a twist, but in this case, this is a Revisionist History podcast called the RFK Jr. Problem.
I listened to this.
What's the twist?
There is none. In this case, there is no twist. Okay. In this case, it's just straight old. This is crazy. And in fact, I think I'm trying to. I think this is a mashup of greed and delusion in the end, because what this does ultimately is it profiles RFK's. It focuses on actually RFK's the fact that he detests. He's got a big thing about the rotavirus vaccine. Okay. About how this is an awful thing, and tries
for
to.
Which there's no data. There's no data for that.
No. Well, actually, it goes into detail. The podcast goes in because in his book that trashes Anthony Fauci, the real Anthony Fauci, I think that's the book. Mm-hmm. It devotes apparently I haven't read it, but it apparently devotes significant time to Paul Offit specifically and the rotavirus vaccine, and goes off on how bad a thing this is. And if they try and tease it apart, and because in the book he's got data, in quotes, I got air quotes here, data about all the harm that the rotavirus vaccine causes. And Gladwell and some expert that he's interviewing try and figure out where these data came from. And in fact, you can correlate it with the packaging insert in the vaccine itself, okay? Taking all the harmful effects from the actual vaccine column of the the safety data, okay? Ignoring the control column, okay? It's like an ignorant look at the VAERS database.
Yes,
Okay.
exactly.
So,
My God.
it's actually, the episode is a very good, and I would recommend it to anybody who might be wondering whether all of this pushback on RFK is legitimate. Okay, because we've talked many times about how the most effective arguments for many people, in particular those who are conspiracy theorists, are anecdotal things, stories, okay? And this kind of fits into that category. This is a description of RFK's shtick, all right, that doesn't rely a lot on statistics and stuff. It's a story, okay? And it's actually quite an effective takedown. It's very good.
All right,
It's worth
good.
listening to. Just a half
Thank you,
hour.
John. Yep, I like Gladwell. It's
Yeah,
really-
and Gladwell likes us, if you'll recall.
Yes.
Okay. So Malcolm.
Hi, Malcolm.
Hi.
Yes, thank you for that. All right, that's TWiV 1211. You can find the show notes at microbe.tv/TWiV. You can send your questions, your comments, your picks, TWiV at microbe.tv. And if you enjoy these discussions, we'd like to have your financial support, microbe.tv/contribute. Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
You're quite welcome. And let me sign off with the opposites of the three poisons. And they are generosity, love, and wisdom. I wish you all three of those.
This administration has none of them. Zero. Alan Dove is at alandove.com, turbotplaque.com. Thank you, Alan.
Thanks. It's always a pleasure.
Brianne Barker is at Drew University, bioprof, barker.bsky.social on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.